In this third tutorial, we'll have a look at using scNodes to correlate super resolution fluorescence data with cryo-electron tomography data. Now, generally, you can use the correlation editor in scNodes to correlate any two types of data, MRC, TIFF, PNG, various file types that we can add. But for now, we'll look at super res cryo-clam. And this tutorial is going to be a little bit more involved than the previous two ones because it involves lots of steps. So before we get started, let's have a look at the final product. In the node editor, you can go to the top menu bar, click editor, and then go to correlation to switch between the two editors that scNodes has. Or you can just press tab on the keyboard to swap. And then in the node editor, I've already set up a project just now, which we can load. Here it comes. And so this is a correlated super resolution and cryo-EM image. It contains 7 different images right now. I'll hide a couple of them. And basically the way it works is that you somehow need to bring your EM and light microscopy data together. And in our experiments we do so by using the reflection from the holy carbon film in our samples, or holy gold actually, and the transmission image, overview image that was acquired in the Arctica in this case. We align the two different modalities based on that, and then we zoom in with higher and higher magnification. So up first is a search image, which is also usually automatically output by the Arctica when acquiring tomograms, and then that's overlaid with an exposure pre-image, and then on top of that, so this is the exposure image, and then on top of this there is the final tomogram, which we can slice through. And then on top of the tomogram is the single molecule localization map. And in this case that would point us to this region and this region of the sample where there should be fluorescent label. These samples are HeLa cells with IMPDH2 labeled with RSEGFP2, a blinking green fluorescent protein. And IMPDH2 tends to sometimes form a filament, which is what we were hunting for. Now in this particular case we were a bit unlucky because, as you can see, there is no filament there. But still the tutorial shows the principle of using the correlation editor in scNodes. But do mind the disappointment at the end of there not being an actual filament. So with that, let's get started with first the light microscopy data processing and then the EM data processing. We're actually loading it all into the correlation editor because we do the tomogram reconstruction in IMOD and EMAN, and see how to set up a scene like this. Okay. If we're just going to do a point spread function reconstruction like we did before, we can use the node setup that we saved in the previous tutorial to save us some time right now. So go to the node editor, file, load setup, and then find it. I saved it on the desktop. So there you go. But now we do need to mind a little bit more which images we register to which other ones, in order to keep our coordinate system compatible with the correlation that we'll use later on between the light and electron microscopy datasets. So we still have this reflection image at the very start of our... Oh, sorry, I'm actually looking at the wrong dataset right now. So I'll open up the one that I've prepared for this tutorial, which is here. Again, we have a reflection image at the very beginning, and we always take these on purpose and throughout the acquisition as well, in order to be able to register our fluorescence images to the reflection image, which we can correlate with EM data. So what we will want to do is add this image to the correlation editor. You can do so by right-clicking and then pressing Add to Correlation Editor. And if you press Tab to switch into the different editor, you'll see that it shows up there. You can also go to File, Import Data, and then select any file that you would want to import. But now we've put this here so that when we get to the EM data correlation, that we've already got the reflection image in the scene. Then back to the node editor. We would want to use not this frame, because this is a UV activation image, which we will get rid of later, but this frame, so the first fluorescence frame that was acquired after the reflection image was taken, this is the one that we will want to register all of the other fluorescence frames to, because this one, by acquisition, is internally registered to the reflection image. So first let's get rid of the reflection images and the UV fluorophore activation images that don't contain the information that we want for the PSF fitting. So if you go to one of the fluorescence frames, in this case you can see that the frame is titled blah 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 channel 5, and the other ones have a different title, so we can use a filter there, channel 5. Title must contain channel 5, filter, and then only those are remaining. So this first one has got to be the template, so press select 0 for the template frame in the registration node. Use the same method as before, which is a bit glitchy at the moment, and then what you can do is move this ROI to maybe select a bit of the image that is better used as a template. So here we still do see the focus drift and focus correction, but the XY drift is better with the ROI set there. So that's all fine. Same filtering as before, same particle detection as before. We could use an ROI again, because we're not interested in the particles that are found in these holes over here. However, the size of the final reconstruction will also change if you do so, and that will make it just a little bit trickier to correlate that reconstruction image with the reflection image, because they won't have the same size. So you have to find where in the reflection image your particle detection ROI was. And you can just use a crop image filter at the very beginning if you want to avoid that problem, or process the entire thing, which we'll do for now. Then let's go for all frames and press go. Okay, so that's done. Took about 2 minutes and we found 1.2 million particles. Let's see what it looks like. And actually seeing what it looks like is a bit easier in the correlation editor as well. So right click, add to correlation editor, and then let's dive into the correlation editor. I'll remove this screen for now and full screen that one. Now we've got the reflection image on one side and the single molecule localization map on the other. And they belong together because they're in the same coordinate system. So what you can do is move one on top of the other, and if you drag it by any of the corners, and then when you get close to any of the other corners it will snap right on. And these have the same size and the same location right now. Not the right names yet, so what you can do there is select the frame and then go to the frames in scene window at the top right over here, right click, and let's call this one cryo-SMLM, and then the other one for single molecule localization microscopy, and then the other one let's call it reflection. Now we want to see both, so let's select the cryo-SMLM one again and go to visuals. Let's make it green, and then at the bottom here you have an alpha slider which you can use to overlay stuff, but what's better is to use the blend mode, and if we set it to sum then we'll have a composite image. So this is the super resolution image within the context of the reflection image. So we can see the outlines of cells here, we can see the kind of the blotting front, where in the center the sample is well blotted and thin, and then beyond that front there is still a lot of liquid remaining, or ice in this case. And if we zoom in onto various holes, we can see that there is some interesting fluorescence going on in that hole. So what I've done is taken this sample to the cryo-EM, I've tried to find the same location, and then I use a map like this one to target holes to acquire a tomogram in. I've acquired 5 tomograms for this square. I don't exactly remember where all of them were right now, but we'll look at this hole over here in a bit. For now, this is fine. Let's make the position of the super resolution image dependent on the reflection image, because that one is in a sense the parent of the other one, and we'll use that parent, the reflection image, to correlate with the EM images. So we can go to the frames in the scene window again, drag the cryo-SMLM tag and move it onto the reflection to make it a child of that image, and then we can lock the bottom one. Now if we select it and try to drag it around, that no longer works, so it's locked in place to the reflection image, and we can select the reflection image, and we have to fix that bug, but not right now. So this is it for now. Next up is adding the electron microscopy data. I'll make this one smaller again. And now I have the EM data in a folder over here, and it contains the, well, one of the final reconstructed tomograms, and also the batch folder that the Thermo Fisher tomography software always outputs, which contains all of the overview images and search images and pre-exposure images. Now the tomogram I've got is Site 2 tomogram 5, so we'll use those search and exposure images as well. But first we'll add the overview image, which was taken at the same region of the sample as the reflection image. So we should be able to identify structures in both this image and the other one. Full screen again, and then we'll undo this for now. Let's just hide the cryo-SMLM image. We can move that frame again. When I show it, because it's locked, I can't move it. It's a bit inconvenient, but I'll fix that before the final release. And as you can see, there is a kind of pointy structure over here, same over there. So let's move this one on top of, right mouse click, send to top, on top of the EM image, and then make it kind of half transparent. You know what, actually let's make it non-transparent, but set the blend mode to not apply. Kind of gives you a view of both of the hole patterns. So first of all, it should be rotated. Second, and I just remember this, maybe you don't see it, but it also needs to be flipped. And then this hole over here probably corresponds to this one over there. So let's drag this one onto the other one. And then zoom in a bit. And set the blend mode back to normal. You can use the number keys, 1, 2, 3, 4, 5, 6, to select each of these blend modes. So by pressing, let's say 4, you go to subtract. Pressing 1, you go back to transparency. Right, so now half transparent, and you can see that the whole pattern is good overlap. Let's move it a bit better. So this one hole is centered, but the other ones aren't because the rotation doesn't match. Now to rotate the image relative to this hole, you can move this marker, the pivot point, into that hole, try to center it a little bit, and then rotate the reflection image like this. Now the alignment in terms of angle seems fine, but the size isn't entirely correct. This is because in the node editor we've set 64 nanometers per pixel for the light microscopy image, but it is actually somewhere on 66 and a bit, so we need to scale it. There you go. And now these two are reasonably well aligned. So, now because we had already set a cryo single molecule localization microscopy image as a child of the reflection image, all of these edits that we've done to the reflection image, which I'll also lock by the way and make a child of the overview, and also I'll move the reflection image to the bottom. Because the cryo-SMLM image was a child of the reflection image, all of the edits to the one have also been applied to the other. So now if we make that one visible again and move it to the top, we'll have and also actually horizontally flip that one. I just horizontally flipped the reflection image, but I should have used horizontally plus children. Then this wouldn't have happened. But there you go. Now we have our EM overview image correlated with the super resolution microscopy image. So up next is to actually get that tomogram in there. So again, we'll go to the batch folder, and you can simply select the files that you want to open in the correlation editor and drag them in there. Or again, go to File and then Import Data. So you can see the new frames appear in the scene as well as in the frames in the scene window. Let's hide the exposure one for now, because the first thing, and also the super res one, because the first thing we'll do is align the search image with the overview image. Now I remember that it was this hole we were talking about, so these two images should correspond. Whenever I actually acquire these tomograms, I always make a note somewhere, or I take a screenshot of this editor and then draw which holes I took the tomograms in. So it can be a bit tricky to find those back. But these correspond. So I can just, and I've set the blend mode to sum by pressing 2. I can drag it roughly into the right position, and then there's a plugin. If you go to the tool window over here, plugins, and select register grayscale, this is a plugin that helps you align one image onto the other as long as they're both grayscale images which contain the same feature. So in this case the child frame is the search image over here, and then the parent should be the overview image. Just translating it seems fine, but you could rotate, you could scale, but for now let's just translate and then press align, and bloop, there you go. It's now aligned with that hole. Then let's get the, let's lock it, and let's get the exposure image in as well. So make it visible again, drag it onto the search image. If it goes behind, right click, send the top. This feature over here kind of allows us to align these two. So again, blend mode, let's see what works. Blend mode, retain minimum seems to work quite well, which is 6 on my keyboard. And you overlay these two ice blobs, blend mode normal, and then again you can use the register grayscale plugin. Set the parent frame to the right one, and then there it goes. So finally we need to import the tomogram, which is in the same folder. There it is. This one happens to be flipped. Depends on how you do the reconstruction in iMode, whether you swap the Y and Z axis or flip it along the X or whatever. Just flip it vertically. And let's see if we can find that same ice blob again. Actually, I remember I should also flip it horizontally, but you'll see that in a minute. Down here you have the slicer, so you can move through the stack and check wherever. You can also right click it and increase the number of slices that you want to sum together, similar to the iMod slicer. And I can see that same ice blob here again. So right now I'm doing this all a bit quickly for the tutorial, so the alignment is a little bit more rough than it would usually be. But let's put that over there. Blend mode back to normal. And now we can just hide the search and exposure. Let's call this one Tomo, side 2, Tomo 5, or what was it? Make it a child of the exposure image. So now everything is parented to the overview, and when I move that one about, everything will move with it. And let's move the cryo-SMLM image back to the top and make it visible. So now what we've got is a tomogram correlated with super resolution fluorescence data. Let's see if we can actually find the protein that these fluorescent labels should be pointing at. We'll select the slicer over here, or what you can do if the image that you want to select is behind other images, because right now if we click here we select the cryo-SMLM one. You can just left mouse click and hold it for a while, and then it will open a menu with all the frames underneath the cursor. So now we're looking for Tomo, and let's see. And so again, there's no filament here right now, unfortunately.